And Masood is going to give us an update on the epidemiology in the Western Cape.
Thanks, Prof. Mankies. I'll just go ahead and share my screen. Is that fine? Can you see everything?
Yeah, seeing that clearly and we're hearing you. Thanks.
Cool. Good afternoon, everyone. So I'll just run right into it. I first want to briefly look at the national trends for the country. So we initially saw increases initially in the Gauteng province earlier a few weeks ago, which was initially focused within the city of Tshwane, but has subsequently spread to different parts of the province. And you can see this clear, steep increases in cases in the Gauteng province in the top right graph. We are now seeing cases and the sustained increase in cases in the rest of the provinces in the country, except for the Northern Cape. Gauteng does seem to be almost at that trigger point to kind of indicate a new wave, and we'll likely see these increases across the province, across the country, indicating a new wave for the rest of the provinces too. If we focus in on our provincial resurgence overview, so this is for the Western Cape, we saw the earliest detection of the new variant of the Omicron variant on the 17th of November. And since then, we can see that the cases have rapidly started to increase, with our 7-day moving average having crossed over our 14-day moving average, and this gap between the two increasing quite rapidly, and kind of making this quite a large gap between, showing this rate of increase to be quite rapid. Although it is important to note that our case numbers are still relatively small, but we can see this clear increase in cases based on the 7-day moving average and also based on our daily caseload, with this private sector predominance that we also saw in the beginning of our second wave and the beginning of our third wave. We're currently seeing 160 new cases daily based on our 7-day moving average. Our proportion positive has also increased rapidly now to around 10% positivity. Our admissions have remained quite low, but we can see this increase in our 7-day moving average in the recent week. And we are now seeing around 11 admissions a day. But our deaths have remained quite low at only 1 death a day on average. So just kind of zooming out and looking at the full picture of all 3 waves for the province from the beginning of the epidemic. The nadir interwave values between the second and the third waves, and also between the first and second wave, and then between the second and third wave, were above what we saw in mid-November, which was around 40 cases a day. So our interwave, if we are calling this the 4th wave, our interwave period was far lower than what we saw in the previous 2 waves. And certainly we can see this increase, even though this graph is really zoomed out, you can see a clear increase in cases in the last week with this 160 daily new cases. For admissions, our admissions have remained low, and if we just compare to the nadir periods between the previous waves, we are still seeing fewer admissions than what we saw between the first and second wave and between the second and third waves. And similarly with deaths, our deaths have remained quite low, and noting possible effect due to vaccinations, but also other things such as clinical improvement in clinical expertise. If we compare the waves, so if we kind of superimpose the waves on top of each other, so just looking at this top graph on the left-hand side, although the 4th wave hasn't quite reached that threshold that we saw with the threshold of the previous waves, it can't be an absolute comparison, but certainly in the private sector, we can see this sort of steep increase in cases. Although just again to note that we are at a much lower baseline than when we were comparing the previous waves. But definitely seeing that kind of increase in the private sector. And if we look at the graph on the right, which is the wave comparison for Gauteng province, the steep increase in cases in this recent wave is quite alarming, almost vertical line. it's still unclear whether this increase in cases will result in an increase in admissions, since we know that admissions do lag behind case numbers by at least a week or two. But I think we need to monitor this closely, and most likely we are already seeing rapid increases, and we might see this kind of pattern for cases in the Western Cape in the coming weeks. If we focus in on our Cape Metro area, so we have seen an increase in cases across the province and across all the sub-districts. In this last week, we saw a 262% week-on-week increase based on our 7-day moving average. And these are largely the larger increases, the larger percent increases are being seen in the more affluent areas. So Southern, Western, Tygerberg, Northern with percent increases of over 200%. And then also in Klipfontein with a percent increase of 131% week on week. And also just noting the private sector predominance in all of these sub-districts, you can see the private sector daily case numbers are much higher. And then similarly in the rural areas, we are also seeing this increase with the 7-day moving average having crossed over the 14-day, and this gap widening between the two lines, but mostly in the Garden Route and the Cape Winelands area, they are seeing larger increases than in the rest of the rural areas. Just to break our cases down by the age bands, so we can see that in the younger age group, so the 15 to 34-year-old age group, the cases have started increasing earlier and definitely more steeper than the other age bands. But we can see now that in this last week, at least, we are seeing this increases across all the age groups for the province. Our reproduction number is well above 1, so it's around 2 based on cases. It's risen quite quickly and is now higher than what we saw early in both the second and in the third waves. And then kind of just to bring this all together, this is the resurgence monitor, which is on the public dashboard for the Western Cape. And we can see that although our case numbers are low, we are seeing steep increases in cases. The week-on-week percent increase has been over 20% for more than a week now, which officially indicates a resurgence, despite the case numbers being quite low. And we know about the emergence of the Omicron variant, and the Network for Genomic Surveillance have been doing a lot of work in sequencing the data. We see now that the proportion of the sequence of the samples that are sequenced, the high proportion now has been found to be the Omicron variant. Delta dominated during the third wave, Beta in the second wave, and Omicron's likely to dominate into this fourth wave. Just to note that the number of sequences have been quite low, but there is ongoing work to sequence more data to determine the true prevalence of the new variant in the country, and also within each province. So for the Western Cape, as of yesterday, we've confirmed 16 sequenced cases in the Western Cape with the Omicron variant. But to note, more than 80% of cases of the PCR specimens in this last week have noted the proxy marker of Omicron, which is the S gene target failure. So that's been found in more than 80% of our specimens for this last week. So although the sequencing data isn't out yet, it is likely that we are already there with this dominance of the new variant. And then lastly, just to note, from a global level, there have been 371 cases of the new variant globally in 24 countries based on the sequencing data. There are also cases in some countries without a travel history at all, or without any recent contact with travelers, which suggests possible community transmission. And I think it's also just important to know that this data is from GISAID, and based on the assumption that the first emerged case is in South Africa, or was at least in South Africa, and this hasn't actually been confirmed yet. That is all from me, and happy to take any questions.
Thank you very much, Ms. Suda, for that timely update on the beginning of the fourth wave and the Omicron variant impacting our epidemic. So, Ms. Suda, can I ask you, it does certainly appear that the Omicron variant is far more transmissible than prior variants. The data, is there any data to suggest what the magnitude of that increase in transmissibility is?
So we don't actually have that as yet, and I think there's ongoing research to determine whether it's the transmissibility or whether it's the immune escape that's resulting in this rapid increase in cases. It could be a combination of the two, but I think that there is still ongoing research being done on that. But we don't know for sure. Yeah.
Still early days on that, yeah. Thanks very much, Masuda. And there might be questions that come up in the question session at the end of the webinar. So if you are able to stay on, that would be great.
Sure.
Okay. Mark, has Wolfgang joined us?
Yes, Graham is in. And
okay. Okay, so to move on to our main talk this afternoon, and it's a great pleasure to introduce Professor Wolfgang Preisner, who's the head of the Division of Medical Virology and at Stellenbosch University and the NHLS at Tygerberg. So I asked Wolfgang to actually give this talk about 3 weeks ago to just discuss the mutations in SARS-CoV-2 and what the implications of those mutations might be. And at that time, I had no idea how prescient and unfortunately prescient the decision to choose that topic would be, because obviously over the last week, the issue around the mutations in the virus has become extremely important to understand. It was really based on a genetic sequence that caused the global alarm around Omicron, and the understanding of what the impact of those mutations are potentially for clinical and the epidemiology of the pandemic. So Wolfgang's going to speak to us on that on the possible and proven implications of the different mutations in SARS-CoV-2, and really looking forward to your talk, Wolfgang. Thanks very much.
Thank you very much, Graham, for the kind introduction and also for the invitation. When I accepted, I thought, well, I have a few quiet weeks to prepare this carefully and, just also revise what we know so far. And well, that came to an abrupt halt about 2 weeks ago or so when the first trace of Omicron emerged, and ever since I don't think I've been able to spend half an hour on this. It's been absolutely crazy, as you probably all know and also have been experiencing. I just stopped my video to preserve bandwidth. So I am to a degree deviating from that topic. I've sort of shifted emphasis slightly towards Omicron, and thank you very much for Masuda for really paving the way very nicely. So I would like to start with giving recognition to NGSSA, the Network for Genomic Surveillance in South Africa, and it also has members at UCT, Hertesker. And I think it's something that the country can be and must be proud of, in that it is actually funded by national sources, DSI and the South African MRC. It was set up at the time in April. It became operational in April 2020, when the only other large country that I'm aware of to have such a coordinated and organized genomic surveillance network in place was the United Kingdom. And all the other big players obviously also did their own sequencing, but not in such a coordinated fashion. And I will explain a bit later what that means and what the value of this is. So a lot of the data and the slides are from NGSSA. That I'm using. So this is the publication which has, that it describes the network. It's now also almost more than a year old, and includes all the participants and contributors. So I'm from Tygerberg, NHLS, and Stellenbosch University, and the PIs on NGSSA from our side are Professor Susan Engelbrecht and Dr. Tongai Maponga. Susan has retired, Tongai has taken over from her, and we are using a unique method called nanopore sequencing. Well, not unique, but at the time it was Susan who set it up, was the pioneer in South Africa. Fortunately, and that I think goes for a lot that is being done during the COVID pandemic. A lot was built and done as part of HIV and of course also in other areas, TB research. So this is why South Africa is so relatively well prepared for this pandemic, because we've been working on infectious diseases for a long time and very well. So the intention was to use this nanopore sequencing technology for HIV. And then COVID came and it was switched over. And initially, with coronaviruses being a family of viruses that have, which is unusual for RNA viruses, not only a comparatively large genome, it's the virus group with the largest genomes, RNA virus group with the largest genomes to infect human beings. So it's 3 times longer than the HIV genome. And it also has proofreading capability in contrast to HIV. So I think it is fair to say, and it has been admitted by quite a few of the globally renowned coronavirologists, that they had not expected variants to emerge in the way that they are doing this now. But nevertheless, of course, being an RNA virus, a virus altogether, one would expect the virus to change over time as it replicates, and that is a tool. So the first applications were molecular epidemiology to monitor where the virus comes from, where it goes, and how it spreads, which can, taken together with traditional epidemiological data, can reveal, very interesting pathways and facts. And this is the first paper written by Susan and collaborators from Tygerberg, and basically showed that the pandemic arrived into Cape Town from predominantly European sources, and she could track back. So that's a North Italian strain. This is from Spain. This is from the UK, and then also monitor the subsequent spread here down here in the Western Cape. Oh no, I haven't got that picture in. So that is simply molecular epidemiology, not worrying about what these mutations would do to the virus and whether it would change its phenotypic behavior in any way, just using it as a tool, which is, it's nice, but, obviously there is no sort of, it's not attached to any emergency type of thing. And of course, the rest is history. You are aware that we are now a bit more than a year after the emergence of the beta variant of SARS-CoV-2, initially known as 501YV2. I come to that in a moment, which very much struck during the beginning of the second wave, and within a short while took over. And I'm sure that the majority of those listening who were around and working in infectious disease at the time are very vividly reminded of a year ago. It almost feels like a déjà vu, what we are seeing now. And this is the paper that describes the beta variant. Obviously, this is now came out, published almost half a year after that work happened. So what are these variants? So while South Africa experienced and defined the beta variant, the UK found the alpha variant, and those were the first two to be found and also described. And what makes a variant of interest? So that's the WHO's definition. Not all countries use it exactly as WHO proposes, but for the global classification that is being used. So it would be a viral lineage that has genetic changes that are either predicted or known to affect virus characteristics. And these includes transmissibility, disease severity, and escape potential in terms of immune response. that could be after previous infection or vaccination, or diagnostic escape or therapeutic escape. Therapeutic escape is largely a dream for us. We don't have those antivirals and monoclonal antibodies that are being used to rescue the odd president should they become infected with SARS-CoV-2. But they also, of course, and we know that from HIV, that antiviral resistance is a big issue. And in addition to those, there should have been significant community transmission or multiple clusters in more than one country, plus an increase in the numbers or the relative prevalence. So taken together, suggesting that these could be an emerging risk to global public health. And there are several lineages of SARS-CoV-2 that have been labeled as variants of interest, or VRIs, and haven't made it beyond that. And I'm not going to spend any more time on them. And I think it's not a bad way of looking out for potential trouble to emerge. But what we are dealing with, and we were a year ago, and now we were dealing with that again, is a variant of concern. And that basically means it does everything that a VRI does. It fulfills the same criteria, and it is also associated with one or more of the following features: an increase in the transmissibility, or some other way in which it has a detrimental influence on the epidemiology of COVID-19. It may change clinical disease presentation or have an increase in virulence, or there may be a decrease in effectiveness of public health and social measures or available diagnostics, vaccines, therapeutics. So you basically find again these same three criteria, but now they have been determined to occur either alone or in combination with this new viral lineage. So how do we call them? And that is difficult. So I had just gotten used to B11529 when luckily WHO gave it an easier name. There are different systems, and I have to say I'm not an expert in this at all, but I do understand, and I follow the discussions. I understand it is highly complex, and things are also always subject to revision, change, and improvements. So there's the Pango, GIS8, and next strain ways of defining genetic lineages of SARS-CoV-2. And this is examples of such names. So the Pango lineage may be B1351, the GIS8 clade could be GH501YV2, and the next strain clade 20HV2. So, but I mean, you can't, it's a tongue breaker and one can't remember, and it's definitely not possible to refer to these easily. And this is why earlier this year, the WHO's Technical Advisory Group on Virus Evolution, or TACVI, came up with a new system. They wanted to get away from people using the stigmatizing place names or country names for variants. So it was the Chinese virus, the UK or English variant, the South African variant. I'm not sure that we have gone beyond that, given what's happening right now, but the idea was that by using Greek letters, it's easier to pronounce, and it avoids misunderstandings, and it also avoids the stigmatization. And this is where our new names come from. And they have equivalents, of course, in the Pango, GIS8 and next strain names. So, they're basically combined. So what we are dealing with, this, that is shown above, is what we know as the beta variant of concern. And this is what happened. So this is looking at the year 2021. This year, we started off with a third, being in a second wave that was dominated by the beta variant, which then stayed with us. And the thing when looking at these curves is always one needs to be very sure what is percentage and what is ends, what is absolute numbers. I come to these to the differentiation later on, but you want to, it can look amazing on percentage, but it could refer to very small numbers of sequences obtained and analyzed. This is not a problem generally for South Africa. You see, this is about on purpose, about 6 weeks back, by which time we had almost 16,000 sequences on GIS8, which is I think an amazing achievement, testament to a very thorough effort. And you see that the numbers obviously reflect the waves during the ebb period. One doesn't need to, sequence that large numbers. And then as the new wave kicks off, one obviously wants to see what's happening. And here, what becomes obvious is that as we started off with better, the second wave ebbed down, and then the third wave started. And it did start with, again, predominantly better, but it was then very rapidly replaced. And that is in line with what happened around the world with Delta, which in the meantime has been split into different clades, but they're all basically Delta, all these greenish ones that very rapidly displaced and replaced Beta virus. I also would like to stress that during that time, we noticed another one called C1.2, 20D, according to the other nomenclature, which we have been watching very closely over the past few months. The reason being that there was some suspicion that this could, again, based on its genetic setup, this could be the next VOI or even VOC in the making. And I think this is also runs counter to people who say that the alert was raised too early. I think it was just in time, because we did not do that with C12 because it didn't show the tendency. It didn't fulfill the criteria. So it was discussed, it was being watched, but it was not, no alarm was raised about something new coming up here. Oops. Well, and this is now the latest from the end of November. Again, GIS8 sequences, and it's very natural that the ends, of course, are lower because of the end of the previous wave. And then they increase, not necessarily only reflecting the incipient new wave, which in fact they do, but also reflecting increasing sequencing effort. And at this stage, the NGSSA member labs are frantically looking back to sequence positive samples that were received throughout November. So what we are seeing now is sort of backfilling of data from previous time points. So numbers of samples will remain confusing for a while to come. And that is not just a South African phenomenon. This is a phenomenon worldwide that, you think, but, how is that possible? Well, we test retrospectively those samples that had been stored in order to understand when first was this observed in a given locality and how did case numbers evolve subsequently. But what one sees very clearly here is that C12 didn't really make much of a change over time. But here is the new kid on the block, the Omicron virus, which took off. But it is necessary to remember that, of course, initial efforts were very much focused on sequencing the samples associated with several cluster outbreaks in Gauteng, and trying to understand that. So one also enriches artificially for samples that would contain this new variant. All those things need to be kept in mind. Some of you may have seen a twittered graph of the different variant-driven waves taking off in South Africa, and Omicron was sort of early and very steep. But that was percentages. So that one cannot say that after having sequenced 50 samples, of which 40 were associated with the same outbreak, you now have, of course, you will have predominantly that particular variant, because this is what you're looking at. So we will get a much better picture once we have filled this up by actually sequencing everything that has been around. And as has been mentioned already, the great thing, and again, I think that's testament for good laboratory practice, that there was something going on, was picked up by an unexpected high rate of the so-called S gene target failure or S gene dropout, which refers to one particular PCR assay, which is used widely in the NHLS and also in the private labs, the TACPath assay that previously with the alpha variant in the UK, already by chance was found to miss in one of its viral targets the variant. So it can serve as a easy and quick screening method for the variant. And this is exactly what's happening. Unfortunately, the NHLS during the trough received so few samples for testing that using the TACPath assay, which many labs have, became not efficient. So we basically used other methods instead, and this is why we didn't pick it up initially. We are now also using that to assess the samples received during November, and if necessary, we'll go back even further to see whether we would already have had the Omicron sequences right then. The private labs, by the nature of their clientele, were receiving larger numbers, and therefore for them it was a good way, a good method to use during that time. So that is one thing to keep in mind when using these screening methods that, of course, in no way replace proper sequencing, but may serve as a relatively quick marker to monitor the spread across time and space of new variants, if they happen to show certain characteristics on the particular assays. So Omicron is the latest addition to the 4 VOCs, variants of concern, Alpha, Beta, Gamma, and Delta. South Africa has now firmly left the Delta period, the era of Delta. And I think we are full on track to enter into the Omicron period. Whether that is something that other countries will follow suit is, remains to be determined. So this is the mutation profile only of the S gene in Omicron. And one has to say that it was absolutely the right thing to do to raise an alarm here, because no other variant has this multitude of mutations in the S gene. You know that this is important, as this is what the virus uses to attach to the target cell, receptor ACE2, but also the main attack point for the immune system, and of course is what the currently available vaccines consist of. So this is obviously important in many ways, and unfortunately, I will just go into a few more here, a bit more detail about the spike protein. So between 45 and 52 amino acids are changed, including some deletions compared to wild-type virus across the whole of the genome, and of these, around 30 are in the spike protein alone. So that is an exquisitely high number. The Omicron variant does not possess a change in the RDRP that has previously, that is present in the Delta variant, and which is associated with a decrease in CT value. In other words, with an increase in viral load. That said, the samples that we are seeing currently have low CT values and thus presumably high viral loads. It will remain to be determined whether this is simply due to the fact that we are now in the upswell of the new wave, where CT values are lower than they are during the peak periods, and later on as the wave comes down again. And that is to do with the point in time in an individual infection during which one samples the patient. So in other words, when the numbers are low, you're more likely to encounter a patient who is actually has all but recovered from COVID, and is now being screened for pre-admission in hospital and so on. Whereas as numbers rise, and they are rising rapidly, as many people can tell just from looking around them. So our lab was affected today with several people being found positive, and then CT values are usually low. Whether that now exceeds what we've seen previously in different waves, and then obviously one can also not necessarily compare the CT values between different assays. So that remains to be studied in more detail. But this characteristic that Delta has is not found in Omicron. That accounts for lower CT values. But it does possess, as mentioned previously, a deletion in the spike protein, which is responsible for the S gene target failure, as also previously found in the alpha variant, which is, just happens to be a very convenient early screening marker if the right assay is used. As an aside, and actually the person who within NGSSA directs the national effort to look at diagnostics is probably also participating in this webinar. That is Professor Shao from Grooterschuur in UCT. He is the leader of the diagnostics group and been doing an excellent and very hard job throughout the pandemic, making sure that the diagnostic assays, and that's not only the different PCRs, but also the antigen tests and ultimately also the antibody tests, are able to detect any new variant that comes our way. And so far, I think the overwhelming news is good news in that there is no evidence of failure of a particular assay with any of the variants. So we don't need to be worried about this, but it's of course always important to look at it, and we are also looking at it now for Omicron. We believe that we know there's no mutations in the nucleic capsid that would affect the sensitivity or ability of rapid antigen tests to detect the new virus. That said, there are sort of reports of people being negative by rapid antigen test, yet positive by PCR, and I think that needs to be studied more closely. And for that, we need not only clinical samples, which is not so easy in that these antigen tests and PCR tests, for the antigen test, you can't just use what's left over from a PCR, so one would have to take a dedicated swab. But what is also going to be very useful is inactivated cell culture supernatant. And along some other labs, we also at Tygerberg have inoculated cell cultures and are busy obtaining an isolate, which can then be characterized, confirmed to be what we think it is, make sure that during isolation and passage from in cell culture, no additional mutations arose or were lost, which does happen occasionally. So they need to be sequenced again to confirm that, then are quantified, inactivated, and can be used to validate different diagnostic assays. So this is just a graphic representation of these different variants or variants wannabes like C12, and this shows where we are with Omicron. And you see there is a lot. It has the same that are found in other relevant viruses, VOIs and VOCs, and it has a number of additional ones, which is of which we don't know the rule. And what we also do not know is in how far they have synergistic or anti-antagonistic effects. So, how these different genetic changes play out in a phenotypic manner remains to be determined. And that is one of the avenues that is being followed by different research groups around the country. So we know that there are multiple changes within the two immunogenic regions in S1, the NTD and the RBD. The RBD is a receptor binding domain, and that includes an amino acid insertion. There are mutations that have accumulated surrounding the furin cleavage site, and including this combination here. And but we, so those are known to be, highly likely to be problematic. Others are known to have deleterious effects from the previous variants, but most changes in the spike S2 subunit, we don't know. We don't know what they do. So that will take a combination of live virus assays, basically working with isolates of Omicron, but also using defined models where you can basically dissect and look for a specific genetic modification and its effects. So the behavior of Omicron, and again, that's been shown in the previous talk already. This is September. This is October. You see the low numbers of sequences countrywide, simply because we didn't have many positive cases. So we were in a position to sequence, to sample a much higher proportion of positives than previously, but the numbers were just very low. It also shows that we are unlikely to have missed anything, but you never know. It will have to take off in a certain way in order to not to escape detection, and then come November. 3/4 of sequences on GSAID so far of the Omicron in Delta is busy being replaced. That said, there's been low activity of Delta before, which means we cannot really say whether Omicron is actively outcompeting Delta or whether Omicron is actually only up against a population with a substantial proportion of some degree of immunity against previous variants and vaccine, which it is, but it's not unlike in the previous waves. It's not that the new coming VOC is actively replacing, i.e., outcompeting the previous one, which gives one some indication as to its propensity to spread. So these are studies that still remain to be done. They cannot just be done by interrogating epidemiological data, and that will require again a combination of laboratory assays like replication assays, where you compare the replicative capacity and speed of different variants in the same cell culture model, but also require looking at certain scenarios that happen in the population. So these are the detection rates, and you see that with surely some allowance for an enrichment, a bias towards Omicron samples. I think the picture that is emerging is very clear, and we are in the Western Cape following the Gauteng in terms of watching Omicron take off in a major way. So these are the figures from the Western Cape. This is the ends this time on the top, and the percentages on the bottom. And you see that even if one assumes that this is not a fair representation of everything that's around here, that Omicron is the one that very rapidly became the majority variant also in our province. So I checked this morning on GSAID, and now just a quick word on GSAID. That is a database that was set up about 15 years ago in response to Indonesia refusing to share information, including sequence information on their highly pathogenic avian flu H5N1 samples from human beings and from poultry. Saying that if these sequences are shared, they will be used by the rich countries to make vaccines, from which Indonesia won't benefit. So GSAID has a code of behavior. One needs to register to access, and one is obliged not to grab and go and do your own thing with the information there, but give acknowledgement and so on. In how far this is working out well with COVID, I think that we will have to look into. So some people complain that they can't easily access these sequences, and others complain that again, there's little recognition being given to the people who generated them in the first place. So that's the background to GSAID. So the first sample to this date, and remember, this is going to be followed up still. So I only looked at first samples in per country. The first one in Gauteng was on the 8th of November, then Botswana, the first one on the 11th. The first one in the Western Cape was found in a private lab sample that Tongai Maponga sequenced earlier this week, again looking back on from the 17th of November, and these are, remember, these are not not the dates of the sequence, but these are the dates of the samples, so they only emerged now, and you see that the list is growing rapidly as countries are conducting their own look back exercises, and I think South Africa deserves a lot of praise for sharing at an early stage when it became clear that this has not only a large number of genetic changes that warrant concern, but also has is spreading very rapidly in Gauteng. Because without that, many of the other countries would not have looked for it, or would not be looking for it now, and it would have taken another week or two until they woke up to it. So this is the current map of the world. Botswana and South Africa are the only countries recognized as having local transmission. The recent case in Japan has only been to Great Britain, so must have become somewhere infected somewhere in between, and there are reports of people returning from Israel and in other countries that had not been to either South Africa or Botswana. So it's very preliminary and it's subject to change, and I'm sure it will. So far we do not know whether these earlier samples reflect the area of origin of Omicron. It might well be somewhere else from where it got to Gauteng and then spread. And as we all have seen, or are still seeing, hopefully not for much longer, behavior had been much relaxed. So I think the conditions were rife for an infectious variant to spread. And you will have seen this, it's lovely, isn't it? We are being locked out of the rest of the world, but actually Omicron is already there. And I also like this one, that is a South African variant. So where next? Well, I think I can report that NGSSA is meeting several times a week to discuss interim results, the latest data, and also plan experiments. So the labs are all gearing up to get their pseudoviruses adapted to Omicron to obtain isolates that mean to be grown up, quantified, inactivated, and so on, which is for translational aspects, looking at diagnostic assays and so on, but will of course also be used for replication assays and for neutralization tests. And there are good banks of vaccine and convalescent sera that will very rapidly give some indication, even though it's only in vitro data, is not clinical protection, and we know it doesn't take into account cellular immunity, but there will be some degree of correlation with protective levels, particularly when one then compares with titers that neutralize other variants of SARS-CoV-2. And then, of course, the epidemiologic clinical observation studies are in full swing already. And again, I'm absolutely impressed as to how much expertise and facilities and everything comes together so rapidly. It's fantastic. I mean, it shows that we've been there before. We are there again now. So we will have a rapid gain of knowledge about this new variant and what it does in practice. And so this is what we are seeing. This is now the isolate, but so it is there, and we are not the only ones that have it. And many countries also want it from us to conduct their own laboratory experiments, and together those two will yield much more information. So the big question is always why South Africa, and I think there's of course a lot of angriness about the consequences of being hit by travel bans again, and we can just imagine how many months those will persist. So why South Africa? Is it a problem that we are just looking too hard? Well, I don't think so, because I think the effort is great in that it provides an early warning to others. So one gains potentially very precious time dealing with this, but I do not think that something that is a variant of concern and is able to spread will be held back for good. So in other words, some of the countries with good and harsh quarantine systems in place where all arriving travelers are quarantined and tested repeatedly, like Hong Kong, would have found it, and they sequence all those cases. In any case, they would have found the variant, and they would have been able to trace it back to South Africa very easily. So I don't think the idea can be to stop looking, and you will then not find there, but it will only be a question of time until it comes out. I think and it's an unproven hypothesis, is that perhaps if it indeed originated in Southern Africa, which we don't know for sure, it could be linked, theoretically at least, to patients with advanced HIV disease that are not on good antiretroviral therapy. And this is the only case that I can find, and it's still in the pre-print paper on medRxiv. It's from KwaZulu-Natal, and it's a patient who had persistent SARS-CoV-2 infection for more than half a year. And you see here the viral loads for HIV and for SARS-CoV-2 over time here. And this is then when the antiretroviral therapy came in, and both the HIV viral load obviously declined, but also indirectly SARS-CoV-2, because the immune competence of the patient was restored, and he was able to control SARS-CoV-2 infection. And during that time, importantly, repeated samples were taken, and they show an evolving pattern of mutations and genetic changes. So in other words, being unable to mount an immune response that would clear the virus, as it would happen in immunocompetent people, such profoundly immunosuppressed people will exert some degree of selective pressure on their SARS-CoV-2, similar to, of course, HIV infected patients on suboptimal antiretroviral therapy. So it is just enough to drive virus evolution and to advantage viruses that emerge that happen to have some genetic change leading to a reduced susceptibility or increased ability to evade the immune response, for example, but not enough to clear it. The same phenomenon has been shown abroad in seriously immunocompromised patients for reasons other than HIV infection. So it does happen. Those, as far as I am aware, all finally perished. They all died. HIV patients can recover if treated. And in fact, we have, I can't show this yet because this is too early stages, but I'm aware that a similar case has been seen at Grooterschuur, and another one has been seen at Tygerberg very recently, where again we have evidence of prolonged active SARS-CoV-2 infection. In the Tygerberg case, probably starting late last year. So that patient was infected for approximately 1 year already with SARS-CoV-2, and again accumulated different mutations. And her virus is a beta, hypermutated beta. I have to say, this is still, it sort of makes sense, but we have not found the smoking gun, and one has to be very careful how to phrase that, again, to avoid repercussions. I think it's a sensible hypothesis. It clearly is a call to look for these cases and to make sure that patients are treated optimally in terms of antiretroviral therapy, and that sorry, forgot what I wanted to say. So that's why antiretroviral therapy. Oh yeah, and of course the other point is HIV patients might need subsequent booster doses of the COVID vaccines as well, in order to elicit the best possible immune response. But there hasn't been a smoking gun. So in other words, there isn't a patient known that has sort of the progenitor of the Omicron virus. From what is known so far, the Omicron is not a variant that emerged out of Delta, so it's not a daughter of Delta. It does not appear to have the Beta variant as its ancestor, so it's not the grandson of Beta. It dates all the way back. It is not clear where it has been all that time. One might speculate again that someone might have become infected early on in 2020 during the first wave, perhaps, and been chronically infected, and over that such a long period of time accumulated, gave the virus the opportunity to accumulate these different changes. But this is entirely unproven. And, I just think it's an interesting hypothesis. It has a few lessons in, these cases, but, we cannot confirm it at that stage, but it might explain why South Africa again. That was it. I'm sorry for talking so long, but thank you very much for listening.
Thanks very much, Wolfgang, for that excellent overview and really up to date, kind of filling us in on what we know and what clearly many things we don't yet know about this variant and the mutations associated with it. I've got one question, Wolfgang. What I find quite surprising is that the backbone of this variant was not Delta and it was not Beta. It's way back to the ancestral strain that was first circulating in South Africa, last year. And yet we don't find any intermediates. So we know that ancestral strain was there last year, and suddenly this virus with that backbone pops up with 50 different mutations. Where do you think the intermediates are? You're on mute.
Thank you. I'm unmuted. Super. Thank you very much. That is the million-dollar question for me. I think this is really intriguing. Where would they have been? How did this virus come about? I don't think one knows so far. I think these cases, and we are still busy, and I believe the same is true for the UCT case, investigating them more closely to understand that better. That is a hypothesis, but I don't know. It's the same question that one has with flus and other viruses that don't cause chronic infection usually. Where are they? If they can't jump from host to host, they don't have anywhere to hide, except if they are long-lived in some environmental reservoirs, like the enteroviruses or so. But those envelope ones, they need to live inside us in order to be alive, to stay alive. So I don't know, but it's an excellent question. What has this thing done in between? Where has it been? I think one needs to be critical about the sequencing efforts. So of course one misses cases, and there are, despite all our efforts to get samples also from the non-academic NHS labs, from the private sector, there are of course gaps, and we don't have the equal coverage across provinces. I think we're doing, NGSSA is doing as best as they can, but it's definitely not a perfect system. And I just wonder whether an additional issue might be the increasing reliance on rapid antibody antigen tests, because except for the early phase when we tried to set up a system whereby they are then also referred for confirmation to the labs, they are positive, and that is it. It stays there, that no sample is submitted. So, and because that is happening in rural areas, we do have blind spots, and one just wonders what may be hiding there. On the other hand, of course, what amount of effort is doable to close these gaps? So, that's the other big question. Yeah. But I would also imagine that further analysis of these genomes, and also looking back into these earliest cases in our country, but also in neighboring countries, may really help to also trace it back to a bit more towards where it come from.
So Wolfgang, we're over time, but I did, Mark Sandrup has just forwarded me 2 quick questions, and if we could just have quick answers to them. The one was, could it come from an animal reservoir?
I think in principle that is one of those things. Think of the Danish mink. They murdered all their minks because they were infected by humans and they could transmit back, and they during that period accumulated different changes that so yeah, I think one can't exclude that, and I think the range of animals that can be infected is growing. So I would not, I think that's the other big hypothesis next to severely immunocompromised people with long-term infection.
Yeah. And then finally, a question about wastewater surveillance. Could that be used to detect emerging variants?
In fact, yes, and I think South Africa once again is actually quite a pioneer there. And in fact, our colleagues from MRC have found variants. The problem is, of course, it ends up being a huge mixture and very impure, as you would imagine. So I think there are some challenges. But yeah, I think this is a great tool, and in fact, this is being looked at very, very actively. It'll definitely be a good tool looking at how this variant spreads across the geography of the country, where there is wastewater disposal, of course. But I think, and of course, there are also samples that one can go back to. But there are some methodological challenges still to be resolved. But yes, I think that's a great tool.
Great. Thank you very much, Wolfgang. A really excellent overview, and appreciate your time this afternoon. I know how busy you and all the virologists in South Africa are. So we've come to the end of our series of webinars for 2021, and we've had 10 very successful webinars this year. We will resume in the new year. I just wanted to say thank you to everybody that has presented on our webinars. To Mark and Wendy, who've coordinated the kind of central sort of engine room of our webinars on a Thursday. And then to say thank you to the Gastro Foundation, to Project ECHO at University of New Mexico, and Cheryl Valentine, Karen Fenton, Sahana Ranglal, Claire Jeffrey, and Mohammed Johnson for all the work that they've done behind the scenes to make these webinars a success. And just to remind you that most of our webinars, the recordings of them can be found on our Department of Medicine website. So thank you also to everybody who's been a loyal attendee of our webinars this year, and we hope we can welcome you back early in the new year. Over to you, Mark and Wendy.
I think really just to thank everybody for their active participation. These webinars only work when everybody joins and partakes, and thank you for all the questions and the real good insights and comments that we've had throughout the year. So look forward to seeing you again on the 3rd of February.
Thanks very much. Bye-bye.